thank you for attending. And what I want to do this afternoon is discussing with you whether or not, how and when innate immune mechanisms contribute to neurodegenerative disease, in particular Alzheimer's disease. And so Alzheimer's disease doesn't need an introduction as at a center like this, but we all know the number of patients, we know the clinical trajectory. What I want still to emphasize is the devastating loss of the self, very nicely depicted here by William Uttermohn, who was affected by the disease and diagnosed in his late 50s, and over the time pictured and portrayed himself, and you can virtually see by every year of progression how he loses himself. And I think this is the most difficult and the most dramatic part of this disease still for me. Now, when Alzheimer used to study the brains of his one and only patient, Auguste Deter. He used a silver nitrate Bielchowski stain as this, and he very nicely depicted intraneuronal changes and extracellular deposits of protein accumulations. And we now know that these are made by hyperphosphorylated tau. We call them neurofibrillary tangles on one end, and the extracellular amyloid beta depositions. And for really decades, this has been the primary and most important pathological changes of the disease. Now, what about innate immunity? What is innate immunity? Coming out of a pandemic, we now have all realized one or the other in a very personal way that we are constantly exposed to viruses, bacteria, parasites, fungi, and what your innate immune system is doing for you can be visualized in this very simple little movie where a macrophage is exposed to E. coli bacteria. And you can see that he very quickly takes the E. coli on and takes them up and digests it and clears them from the plate. So this is what your immune system does constantly for you, because you have a breakthrough of bacteria into your body virtually every second. And if the innate immune system would not be in place, you probably would not survive a week. You would develop organ dysfunction due to endotoxemia and die from septic shock. Now, the innate immune system represents our first line of defense. These cells do not only exist in the bloodstream, they also exist in the organs as resident cells. In the brain, we call them microglia. In the lungs, alveolar macrophages, copper cells in the liver, mesangium cells in the kidneys, Paneth cells within others in the gut, and bone marrow macrophages in the bones, of course. They all protect these organs from infection, but they also contribute to their structure and their function. And the first time these microglia cells have been experimented with was actually here in Madrid by Pio del Rio Ortega about 100 years ago, who did very elegant experiments at this time. And there's a great review article on the historical aspects of his work by Amanda Sierra, published in Glia some years back. Now, what happens in Alzheimer's disease in a nutshell, very oversimplified, you have a transmembrane protein called amyloid precursor protein, which is cleaved by beta and gamma secretase in a sequential manner into the so-called amyloidogenic pathway, generating amyloid beta monomers. The best known of are amyloid beta 1 to 40 and 42, but there are others. And they should leave our brain by about 25% into the CSF and 25% in the lymphatic drainage pathway. Now, ultimately, the remaining 50% are being taken care of by microglia cells, either through the secretion of enzymes such as IDE or neprilysin, ACE, or by the phagocytic uptake. If that system gets out of balance, we have the formation of oligomers, which show here this shift to higher molecular weight, and ultimately the deposition of fibrils in the brain parenchyma. This Deposition starts an inflammatory reaction characterized by the activation of microglia cells and GFAP-positive astrocytes. I will come to that in greater detail. And ultimately, there is neurofibrillary tangle formation in the neurons, initially indicating the dysfunction and ultimately the death of these cells. Now, when does Alzheimer's disease starts? And about a decade ago, and Michael here must know this much better than I do, one would have said, well, maybe around the time the first short-term memory deficits are being sensed by the patient, but that's completely wrong. This is showing you a sequential PET scan that goes back 25 years prior to the first cognitive symptom taken with the Pittsburgh compound 2. It's an experimental, not a clinical assessment, because these traces are not so long around. Now we go 25 years back, and you can now see over this time how the brain fills up with amyloid beta, the precuneus. You have a sagittal view and a lateral view. The precuneus is nicely to see the frontal parts of the brain. Also the temporal lobe, the mesiotemporal lobe up here. Now we're still 5 years before the first memory symptoms are appearing. If it turns to 0, this is the time point when the patient recognizes for the first time that something is wrong. And now we go 10 years into disease, because in the mean, this disease is being survived in the Western Hemisphere by 12.4 years in the mean. Now, I take 2 things from this video. First of all, if you look to one brain hemispheres, there are hardly 2 brain areas which are precisely in the same stage of disease. This is a spreading disease, which has a phenotype that affects the brain areas in a different temporal manner. And that's important. Another point is that obviously, if disease arises in greater numbers, 65 years of age and older, if you go back 3 decades, this disease, at least from what we can figure and trace today, does not start like later than the 40th birthday. This is the 4th decade of life, which is important, where amyloid beta deposition seemed to start. So we have a relatively long, clinically inapparent, but pathobiologically important stage of disease. Now, this is showing you a cortical section of a brain, a frontal cortex, and stained for beta sheet structured amyloids with Congo red and MHC class II positive microglia cells. So, but long before the brain has been stained with Congo red was used and developed for something completely different. Does anyone know what Congo red was used for? Congo red was developed as a stain for cotton and shirts by Bayer in Leverkusen in Germany, until it turned out that it's a very rainy region of the country. It would be washed out from the clothing. It would not stay in the textiles. So they put it back into, and a cleaning person by accident would throw that vial to the lab floor, leaving the lab floor in a very strange stained fashioned manner. And there was a very clever intern, Paul Bötticher, in 1880, who found out that the reason for this stain would be that Congo red would stain bacteria. And he went to Berlin and licensed it as a bacterial stain. But then he made a mistake, because he tried to sell the patent to another German company, Agva, without telling Bayer. And a big lawsuit unfolded, and that's why we know the story so well. So, but it raises a question. If it stains bacteria, and if it stains human brain, what's the commonality? And it took about 100 years until Matthew Chapman very elegantly showed that bacteria very common to us, such as Escherichia coli, Staph aureus, Salmonella typhimurium, call beta sheet structured amyloids on their surface. Microbiologists call them different. They call them curly fibers or curly operants. And they look very much the same as the one that deposit in our brains. But that means that myeloid cells, such as this microglia cell, over 1 million of years, has been taught through evolution that a beta sheet structured amyloid is nothing else than a pathogen invasion. So what happens is that nature has equipped these cells with pattern recognition receptors, including the toll-like receptor family, CD47, CD36, scavenger receptor A and B, to sense these beta sheet structured amyloids to defend the body. And this is exactly what happens. So the myeloid cells mount a massive reaction, which on one end generates a lot of inflammatory mediators to kill the opponent. And the second reaction is a phagocytic clearance reaction. Now, this happens on the background of our lifestyle. And there are various lifestyle factors that fuel into brain inflammation, including obesity, especially in young men, poor oral health, periodontitis, aging by itself through the generation of a senescence-associated secretory profile of the tissue, brain trauma, very direct, infections and systemic inflammation, as we showed in a very nice joint project, and sedentary lifestyle. You may like it or not, but sitting a lot in front of your computers and not working out increases cytokine levels in the body. This is what we can change. What we can't change is our genetic background. And over the past decade, we experienced the identification of various risk variants identified by genome-wide association studies. And of these, about 96 identified risk variants, 50% directly relate to innate immune pathways. And there are roughly 30% more to pathways involved in lysosomal and autophagosomal function. So very important for microglia cells as well. But none of that, regardless whether you look at BIN1, TREM2, CD33, and all the others, none of them would tell you in which cell these risk variants play their evil game, right? This is just a genetic analysis. So that has been detailed very elegantly by Kozo and colleagues and published in a Nature Genetic paper in August 22. They did is they used 150 patient brains and isolated microglia cells, astroglial cells, neurons to detail in which cell risk variants would actually act. And what they did is they used a single-cell RNA sequencing data set and an attack sequencing approach with transcription factor footprinting and would run the GWAS information against it. Now look to just the red box, please. Parkinson's, ALS, and Alzheimer's. N positive, a role in N positive cells versus the role in microglia. When Parkinson's disease, none of the risk variants seem to play a role in neurons. There's a slight but significant role in microglia cells. In ALS, most strongest signal here for playing a role in the neuron itself. Well, there is a tendency, but it becomes due to the high variability, not statistically significant for an additional role in microglia cells as well. But in Alzheimer's disease, there is even a negative signal for any risk variant playing a role in neurons, while the signal for microglia cells is the highest and massively positive. That proves that this disease is driven by microglia changes. It doesn't mean that it starts with it, but it's massively influenced during the process of ongoing pathogenesis. Now, we all know in the pre-symptomatic stage, I showed you the amyloid beta deposition already in the video, but at the moment you have amyloid beta depositing, you get that microglia reaction through the pattern recognition receptor ligation, because to the microglia cells, it's nothing else than a pathogen challenge. And so for the first part of the talk, I want to focus on this. And here, you can see that microglia cells, which have a wide spread out process network, they retract all these processes, they become round to oval, and they interact with the plaque deposit. They start to release a lot of inflammatory mediators, including complement factors, chemokines, cytokines, nitric oxide, and radical oxygen species. Now, if we start to interrogate whether of these play a functional role in disease pathogenesis, we need to be very careful. Why? Because only a fragment of the adaptive and the innate immune mechanisms are conserved between mouse and man. Roughly 70% of the adaptive immune system is not conserved, and 60% of the innate immune system is not conserved. So that's why it was very important to us to look at this data set by Eric Bodeke's group, published some years back. And Eric and his team took 4 different transcriptome data sets from human cortical microglia cells, and they were defining the overlap, the core signature of human microglia in the disease. And in the core signature, you find here the inflammasomes, especially NLRP3 and PICARD. PICARD stands for, is an alias for ASC. I will call it through the rest of the talk ASC, but PICARD is the alias. And the products here on the cytokine side. Now, the NALP3 inflammasome is a tightly regulated system because it mounts a profound inflammatory response. It requires the ligation of a toll-like receptor or cytokine receptors and the upregulation of proforms of cytokines and auto-inhibited NALP3. The next step then, and if there's no further stimulus to that system, it will stop here. It will not execute inflammation. It will not react. If there is the re-exposure to an inflammatory stimulus or mitochondrial damage, lysosomal damage, potassium shift across the membrane, the buildup of the active inflammasome is licensed and executed. And this forms a wheel-like structure. And then these wheels are being put up. Imagine a tire, and just imagine you would put all the tires of your car on top of each other. So this is how an inflammasome looks like. And here you see the outward directed majored ASC spec with the caspase activity, then cleaves spermine D. The cleavage product moves to the membrane and forms a pore complex. The active caspase 3 cleaves pro-Al1 beta and Al18, and this mechanism then leads to the coordinated release of these cytokines into the extracellular space. Now, this system is on in Alzheimer's brains. This is an older paper where we found ASC specs, so activated inflammasomes in microglia cells of Alzheimer's patients, but we did not know how active this would be. That could be strongly or weakly induced. So we developed a system where we would cleave for caspase 1, so blood for the pro-caspase signal and the cleaved caspase signal as a sign of activity. And doing this in frontal cortex and hippocampus section, not shown here, and this is just the frontal cortex in age-matched non-demented controls versus 80 cases, showed you that there was a strong upregulation in Alzheimer's disease patients, which luckily is being replayed by the mouse models we have. So this is a mouse model generated by Joan Jankowski and David Borchelt already in the late 90s. And you can see in an age-match dependent manner, we have the cleaved caspase 1 signal here. And when we cross in knockout animals, we have the loss of the signal. So this is direct NALP3 inflammasome activity. Okay, so summarizing a really strong and wide paper with more than 30 supplementable figures. In one slide, when we look at the knockout of NALP3 on the APP-PS1 background, we prevent microglia activation. We increase physiological amyloid beta uptake and degradation. We thereby reduce the cerebral amyloid load in 18-month-old animals by 70 to 80%. And importantly, and this is work by my collaborator Martin Korte from the Technical University in Braunschweig, we protect from APP-PS1-induced spine loss. And Martin and his team then went on and looked at synaptic plasticity in the hippocampal Schaffer collateral. And what you see here is that the presence of APP-PS1 suppresses the hippocampal LTP, the synaptic plasticity. And this is completely protected in the knockout animals. And it didn't stop there. Having protected synaptic plasticity of the hippocampus, let us test the spatial navigation memory of these animals. And here we used many tests. I showed just just one data set for time reasons. This is the Morris Water maze test. The animals have to search for a hidden platform in one of the quadrants, and they can only orient themselves at external cues. And you can see that wild-type and knockout animals, they learn the task over the first 4 days, and then they consolidate that information. While APP-PS1 animals hardly learn anything, and the knockout APP-PS1 animals are almost as good as the background controls. Now, at day 9, we remove that platform and we interrogate whether the animals had been following a spatial navigation strategy and build some memory. So we ask whether they would confine the search strategy to the quadrant where the previous 8 days the platform had been located. And what we see is that the wild-type and knockout animals would confine their search to quadrant 1 where the platform had been. The APP mice have a random search strategy, while the knockout APP mice, they confine their search entirely to quadrant 1. So they have a very preserved memory just by preventing the inflammatory reaction. Now, next to this inflammatory release, and also through the continued inflammatory release, the microglia cells will sooner or later die, and they die from a pyroptotic death. So microglia pyroptosis leads to the release of the inflammasome. The ASC speck is being expelled. And this is for a good reason. Imagine a skin wound. In a skin wound, you have invading bacteria. You have the death of your defending immune cells. And if the new immune cells, which come from the bloodstream, would have to go through this long initial phase of priming and licensing of the inflammasome, which takes hours, the bacteria would be much faster and overtake the body. So in order to make the inflammasome ready and usable for invading cells, the invading leukocytes can take up the inflammasome and have a ready-to-use and ready-to-fight inflammasome to defend the body. In a skin wound, it makes totally sense. It does not in the brain, as I will show you. Okay. This is what happens. You see the constant exposure to inflammatory stimulus is leading to increase of L1 beta. At the same time, the mitochondrial respiration inside the microglia cells breaks down, so their metabolism gets weaker and weaker, and the phagocytic clearance capacity also breaks down. And ultimately, these cells die. And this is an experiment where we wanted to show that beta amyloid is a strong inducer of microglia pyroptosis. So in the supernatant, you have TAMRA labeled, red labeled, amyloid beta 1 to 42. And this cell will now undergo a pyroptotic death and release an ASC speck. And the moment the ASC speck is released, the amyloid beta forms a hollow structure surrounding it. And you see this at 2 positions. So when we saw that, we hypothesized that a microglia cell that undergoes an inflammatory stimulation, ultimately dies from pyroptosis and releases this speck, would create an ideal matrix for amyloid beta deposition. So basically what we hypothesized is that we have an amyloid beta deposit not at the site of any random choice. We have a deposit at the site where a microglia cell had been dying before. So we then looked by various biochemical tests, and I show only some. The whole paper has 41 supplementable figures. It went through 4 rounds of revisions and was a long fight. So if you're interested, then go to the paper. This is showing you the thioflavin T assay. It shows you the conversion of a single monomer to an aggregate. And in green, you see the aggregation time and dynamics of amyloid beta alone. And then I hope you can appreciate that adding in ASC specs and increasing concentrations shift to the left massively. And this is a classical sign for cross-seeding. But we didn't stop here. We was not convinced by that assay too much. And here you see in a blood system that after 24 hours, in the absence of any ASC, there's hardly fibril and oligomer formation. This is amyloid beta 1 to 40. But with increasing concentrations of A beta ASC exposure, we would see fibrils and oligomers. And this is the same for 1 to 40 as well as for 1 to 42. Almost nothing in the presence of in the absence of ASC, but in the presence there's strong fibril formation. Now, that made us thinking. And so we thought, better we give this out of our hands because that's really too cool to be true. And so we went into a collaboration with Jochen Walter and his team. And Jochen is really great in doing co-sedimentation assays. And what they did is they looked to A beta 1 to 40 and 42 alone, together with ASC or ASC alone. You see that at time point 0, everything is in the supernatant, while at time point 6 hours, everything has been going to the pellet. So the ASC in the solution has dragged the A beta into a plaque-like structure within only 6 hours. Now that prompted experiments where we looked at mice. And here we see that in an age and transgenic dependent manner, we have A beta by co-precipitation binding to ASC. But more importantly, when we looked to the immunohistochemistry of these plaques in the mice, we found that the core would be made out of ASC, and the 6E10 positive amyloid beta would form a hollow structure around it, very similar to the pure test tube experiment I just showed you. Now, we then thought, if that's true, we should be able to prove that ASC is a seeding element for amyloid beta deposition. And what we employed was a model that had been introduced by Larry Walker and Matthias Jucker, nicely described in this Nature manuscript in 2013. And what they proposed was to inject solvent control or wild-type mouse brain versus mouse brain lysates from APP mice into opposing hippocampi. And in various models, they showed that you would see spread of pathology on the hemisphere that would receive the APP tissue. So we looked at the spread of pathology. This model is important because it has a problem. Synthetic amyloid beta is not seeding. Synthetic amyloid beta has no effect, which means that we either have differences in the amyloid beta itself, or we have a cofactor that is important for depositing amyloid beta. And there's a cofactor for listening to me, which is oxygen, and therefore I open the window. Most of our brains work better with oxygen. Maybe you open one in the back. Okay, so this is the most important slide in the entire presentation. No escape room. Okay, so what we do here is we have APP mice, and in opposing hippocampi, we inject wild-type brain lysate versus APP PS1-derived brain lysate. And what you see is that we completely validate a science paper published by the Yucca group in 2008, where we see that we have a twofold increase in the number of plaques and in the total area of amyloid beta deposition. But we went beyond that, and we looked to the APP processing. So APP processing was not affected. Total A beta was massively increased, and so was the smear of amyloid beta oligomers and fibrils. And I wanted to know how much the injection does. So we looked at non-injected hemispheres, and you see the injection itself doesn't do anything. Here, not at the smear, not at the processing. And now we repeated the same experiment in the absence of ASC. Just a little inflammatory molecule. And you see that aggregation, as in the test tube, and oligomer formation is completely absent. So is the potentiation of deposition. And we concluded from these experiments that then in this mouse model, the spread of pathology strictly depends on the presence of ASC specs. Now, What about humans? Why is that important? Again, in humans, in the AD patient brain, as compared to controls, amyloid beta binds to ASC exclusively in patient brain. And then I had a clever postdoc, and he took a lot of my money and bought about 150 brains from Banner Health, 50 brains from controls, 50 brains from MCI cases, and 50 AD cases. And he used them to do split the core of the plaque from the fluffy fiber compartment by a sucrose gradient centrifugation protocol. And what you see is that in the MCI stage, we already have ASC and A beta in the core. And the only difference of the patient is that we have more amyloid beta in the fluffy fiber compartment. So when we started to stain these plaques, they all look like that. Every human plaque has an ASC core and a 6E10 positive halo. So I thought this is way too good to be true. I don't, believe that. So we therefore went back into cell culture and looked at this phenomenon in further detail. And here we found that we can find the formation of plaque-like structures at 4 different positions. Here, 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 here's a further one, here, where you have the ASC speck in yellow now in the center and the 6E10, the TAMRA labeled amyloid beta surrounding it. At day 4, this is a tiny halo. It grows and it grows until you have a ball-like structure which has an ASC positive center. And we now use this and oops. We're now going to use that further, and we don't like to kill animals in the lab, so we have a 2-photon in vivo setup where mice walk a treadmill, and we can image the brains of these animals while they are alive, they're awake, they're not anesthetized, they're usually happy because they get a sugar treat at the end, and we can image what happens in the brain. So we generated a mouse which is APP-PS1 transgenic, has a CX3CR1 EGFP, meaning all the microglia cells are green, and a knock-in ASC-M cherry. So the videos we generate show you yellow ASC specks in the microglia cells. If they turn red, this is when the cell dies and the ASC speck is being released. And you see when we inject methoxyxo4, it's a blue dye for amyloid beta, it forms these halos. And in the next video, we took the green channel out, and we only have the ASC-M cherry and the blue amyloid beta. And I want you to precisely watch whether you find any deposit, any amyloid beta deposit that does not have an ASC core. There is none. These mice have not been injected. These mice have not been made inflammatory. They were living a happy mouse life. They are not even anesthetized. And every plaque has an ASC positive core. Now, this is a larger magnification here. You see the ASC inside the blue amyloid beta. This is the plaque's environment. You see there's lots of ASC inside and specks inside the microglia cells. And if we do that in a time-lapse manner, we see that the release of ASC specks are increasing while the deposition then takes on. And here we asked ourselves, when this is really seeding, the ratio from magenta to blue should drop because there's more and more amyloid beta attaching to it. And here you have this phenomenon. We have a massive decrease in ratio, meaning that ASC is seeding and A beta is subsequently attaching to it. And this is a time lapse, baseline 3 weeks, 6 weeks, 13 weeks, and you see how it massively increases every time at the site of a previous AskSpec release. So these are large movies. We scan brain 300 to 300 to 700 micrometer constantly over an hour. So it's more than 1 terabyte per movie. And we need to, there was no software which would allow us to compare over time these data sets. So we now wrote a code, and this is available at fairsharing.org. So it's freely available. Everyone can use it and can test it and can run it. So please feel free to work with it anytime you run similar experiments. It allows us to track single plaques over time. And that's important. You see how the blue plaque volume takes on while the red stays down here. It's important because we now will be able to start testing drugs. We developed a system where we have a 3D cell culture, and we induce AskSpec release and the presence of amyloid beta. And you can again see the Ask is the center, and you have this ball-like structure around. So this is in the absence of immune stimulation. This is with immune stimulation. You have the increase in plaque number and size. In the Ask knockout, you virtually have no plaque formation whatsoever. And interestingly, now we can start to target this, and we have identified the site where amyloid beta and Ask interact. So we raised antibodies, and we humanized these antibodies, and we now have 2 antibodies. And here you can see that they both are effective. Antibody 1 is a bit better than antibody 2. And then we went through a whole library of 4,000 drugs, which would all pass the blood-brain barrier. And our question was, which of these drugs would prevent pyroptosis, inflammatory death of microglia cells? And we identified 13, of which we then nailed 5 down, which were highly effective. And you can see this is compound 1 to 5, and especially compound 3 is highly effective in preventing the formation of plaque deposits over time. It's just one deposit at 10 micromolar, and we will follow up because we now use the in vivo setup to test whether these drugs will also work in vivo. What we believe is that we can prevent the spread of pathology at an early time point. Now, when there is death, there is also birth. And microglia cells proliferate in the Alzheimer brain. And we know that from very elegant work by Jonas Friesen from the Karolinska Institute in Stockholm. And Jonas looked at a big human experiment because, as you may know, the land of the free has donated to human mankind supraterrestrial nuclear testing. And each of these nuclear tests conducted between 1940 and the year 2000 within 48 hours leaves the entire planet enshaded in nuclear waste. And this is C14 mainly. And the C14 from these nuclear detonations can be distinguished from each other. Can you imagine how? By the decoy times. Okay, so you have to know that the C14 incorporates in our body. So everyone which is born 90, and older has at least nuclear waste from 3 supraterrestrial detonations in all his or her proliferating cells. Not nice, but a great human experiment, right? So what Jonas did is he went to the physicist at the Karolinska, said, can you distinguish these different C14 fallouts? And they could. So what they did is they calculated that microglia cells would renew at a median rate of 28% per year. So in a healthy human brain, roughly one or more than a quarter of microglia cells is renewing per year. Microglia cells live 4.2 years in average, and most of the microglia population will be renewed throughout life. So imagine if you live 80 years and a microglia cell is getting 4.2 years old in average, you may have 20 generations of microglia cells, correct? Maybe 15 or maybe 10, doesn't matter, but several. So that's important because we hypothesize that it makes a big difference whether a microglia cell proliferates into a healthy brain, into an early neurodegenerative situation, or into a chronic degenerative brain. Microglia cells proliferating here will be like kids born into a war zone. They get completely different cues. They will develop completely different. They will have a different transcriptome. And indeed they do. So what we did is we compared the proliferating cells from wild-type versus APP mice at 6 and 12 months. And what you can see that we get an upregulation of pathways, cellular response to LPS, exchange LPS for DAMPs, danger-associated molecular patterns, regulation of phagocytosis, regulation of neuron death, antigen processing, and regulation of leukocyte differentiation. And here you find many members. TYROP-BP is one of them, IRF, MPEG-1, and others, TREM2, which belong to those genes that are being found back in the GWAS hit analysis. Now, when we analyze this, we see that we lose from 6 to 12 months, we lose the tolerogenic part of the microglia transcriptome, and we acquire a neurodegenerative part. If we then go and analyze the differentially expressed genes, we find that at the beginning, roughly 20% are shared with the so-called DAM signature identified by Ido Amit's lab at the Weizmann Institute. At 12 months, it's already 50%. So what we believe is that the so-called DAM signature is not the individual response from one cell, but it's the population exchange through proliferation. Now, we looked whether these cells would be still fit to conduct their work. And so here you can see that there are no cells which are proliferating and have taken up amyloid beta, virtually zero. There are some which are Ki60 positive or Ki67 negative and methoxy positive. So they have not been proliferating and they took up amyloid beta. Most of them don't take up any amyloid beta, but that's highly statistically significant at 6 and 12 months of age. Now, we looked at the transcriptome of these cells and we found that there are various inflammatory mediators, lots of different inflammasomes, NALB3, AIM2, NLRC4, NLRP1. These are the 4 ASC-dependent inflammasomes. So we started to knock out ASC and looked whether we would regain amyloid beta clearance. And in fact, we got approximately 25% back. So some of the inhibition is dependent on ASC, but not everything. And so we'll have to further dig into that. And maybe that's also something that could be done in human cells. Now, I told you this is just the pre-symptomatic stage. So how we get from here to here, how we induce tau pathology. And to answer that question, and this is Christina in the lab was adapting a paper by Jürgen Götz, a model he published in Science in 2001, and what we adopted this to tau-22 animals, and I can explain why we did so. But we injected wild-type mouse brain versus APP-1 brain lysis into the opposing hippocampy of tau mutant mice to drive tau pathology. And what we found is when we have wild type mouse brain hippocampy versus the APP receiving hippocampy, we see a massive difference in pathology. This is the red bars in comparison. If we, however, do this on the ASC and NAB3 background, we don't see such an increase. So, and to make a real whole paper in one slide for time reasons, we saw that when we have pattern recognition ligation drive interleukin 1 beta secretion, we activate the L1 receptor on hippocampal neurons, and in a MIDI88 IREC4 dependent pathway, we phosphorylate MAC1/2. MAC1/2 phosphorylates CAM kinase 2 alpha, and CAM kinase 2 alpha phosphorylation of tau leads then to loosening the tau from the microtubule, forming paired helical filaments and ultimately neurofibrils. So now the question is, are microglia cells always evil? Is this something we just need to suppress? And as ever in nature, the answer is no. This is a data set where we looked at a system, the so-called TAM receptors. And there are three TAM receptors, Tyro3, AXL, MERTK, and these are receptor tyrosine kinases, and they regulate immunity via STAT. They suppress through the now. Okay, we repeated the entire talk. Not today. Okay, so for the TAMA receptors we could measure at that time, which was AXL and Tyro, you see a massive difference in the healthy controls, which was dependent on the presence of tau. And I personally cannot recall any strong difference as such between in a tau-dependent analysis. This is almost all or nothing. What you can see is that for both receptors, the system seems to burn out towards Alzheimer's disease. It becomes statistically insignificant in the 80 patients and less significant between the groups over time. These results were backed by data from the Fundació ACE from Barcelona, and then we looked at the effects on brain volumetry. And what we saw is that what we expected is that we see a negative correlation, but we found a positive correlation. So it was exactly the opposite than expected. And what happened was that AXL and Tyro were positively correlated with the preservation of tissue in I feel more energy. Okay, good. Back to the data. What you see is that the tissue preservation exactly concern those brain areas which are affected by Alzheimer's disease. Okay. And so it didn't stop with tissue preservation. If you look here, we look at cognitive, this is a composite score, we look at cognitive decline. People with high tyro were more stable and high axle were more stable as compared with people with low tyro and low axle. So at the beginning of the disease, very, very early, microglia release and microglia ligation by TAM receptors would be protective. As more immune suppression we have at the beginning through TAM receptors, as better the tissue and our cognition is preserved. So we were super interested. Why? Because here nature has proven that there is a mechanism which concerns microglia cells and which leads to the direct brain protection and not only morphology, also function. So we looked through all the systems we know to date, and the only modulation we found was the modulation of the inflammasome. And what we saw is that thyroid overexpression would completely abrogate interleukin 1 beta generation, slow down massively caspase cleavage and R1 beta cleavage. So tyroligation, other than axle ligation, is a massive influencer of the uh, and modifier of the NAB3 inflammasomes. We do not know yet what is the axle effect here, but we are sure that tyro3 works through the inflammasome. And that's why we believe that inflammasome compounds have a future in the treatment of Alzheimer's disease. Now, I told you that at the beginning, Alois Alzheimer and Franz Nissel looked at the silver nitrate Biolchowski stain, which we have seen 1,000 times. And that's true. It's true, page 1 of the publication. The following 9 pages depict microglia cells, believe it or not. This is showing you a nice scene of microglial pyroptosis and microglia suffering. Here he called them amoeboid glial cells. That was his way to describe microglia sitting and interacting with a neuron. Here it's an amoeboid glial cell that sits at the vascular deposit, several in and around an A beta deposition. I just think, despite they have made so much effort to paint 9 pages of these beautiful pictures. We just ignored it for more than 100 years. And we better go back and think about the role of these cells. Now, in summary, I hope I could provide evidence that the NAB inflammasome is a key innate immune signaling pathway activated in AD. NAB3 knockouts in the mouse models we have improve A beta clearance, hippocampal LTB, spatial learning, and memory performance. By now, these data have been reproduced at large with using NAB3 inflammasome inhibitors by several of the companies. Ask specs, seed A beta deposition in vitro and in vivo can be targeted by antibodies and compounds. NAB3 derived interleukin drives tau phosphorylation in neurons and pathology, and several companies, Sanofi, Merck, and others, prepare clinical trials in Alzheimer's disease. Most of them will enroll next year. I've been told we are still involved in a co-development of anti-ASK-A beta antibodies with AC Immune and Mabilon, and try to push this also with small inhibitory compounds. And this is largely done by people in Bonn and now in Luxembourg. And I'm very thankful to the great collaborators and, of course, to all the people who pay that work. And I thank you for your time and your attention. And I hope you have questions. This is my battery.
Well, thank you very much for a wonderful talk. Questions?
Yeah, thanks again for this great presentation. I wanted you showed these images of these typical amyloid PET patterns that are reproduced by all amyloid PET tracers. We know it's always the same pattern, very parietal, very frontal, almost complete sparing of occipital of the primary sensory motor cortices. And I wonder whether this nobody really knows why some of these regions are so spared and others are so susceptible. And I wonder whether this whether you know any if there's any data about like normal physiologic differences between these regions in their microglial density, microglial function, or whatever, may be differences that they're just microglia for some reason, some functional reason, some physiologic reason. Some in microglia may function differently between these different regions, and so then also affect amyloid seeding, basic or aggregation differently.
It's a very good question, and I don't have a clear answer, but I have a hypothesis to offer. So the microglia cells, and not studied very well, have carry every receptor for any neurotransmitter you can imagine, and neurotransmitters modify microglia function. Norepinephrine, as one example, is a very phylogenetically very, very old. Before it became a neurotransmitter, it was an anti-inflammatory. So acetylcholine is suppressing inflammation. We know that serotonin is modifying microglia function. So what I believe is that having different neuronal populations in different brain areas will also consequently cause a different neurotransmitter profile. And the neurotransmitter profile maybe make microglia in some brain areas more susceptible to inflammatory reactions as compared to other brain areas. Maybe they make them more susceptible to periapoptotic death as in other areas. That would be something we can test. One of the things we were super interested in for many years is the early degeneration of the locus coeruleus because it leaves the projection areas, mainly the limbic system and the neocortex, depleted from norepinephrine. And we have shown in several papers that this raises inflammation and especially interleukin 1 beta levels. So we now go back using the mouse model we have and deplete the locus coeruleus by a toxin called DSP4, and we will test whether we have a higher likelihood of plaque deposition and seeding in this model. But it will not be restricted to norepinephrine. As I said, there are receptors for glutamate, for serotonin, for acetylcholine, for norepinephrine, for dopamine, virtually every neurotransmitter you can imagine.
Okay, thanks. And the second question was about the very intriguing data about like basically uh, um, um, cognitive of these cognitive effects in the inflammasome knockout APP inflammasome knockout mice. So based on that data, is amyloid overproduction a bad thing?
Yeah, I think as soon as it starts to represent a DAMP, a danger-associated molecular pattern, if it leaves the brain in a physiological manner, it may not. If it's overproduced, I'm I also think if you compare the immunogenicity of 40 to 42, if you look to most of the presenilin mutations, there's not an increase in total A beta in these familiar mutations causing familiar Alzheimer's disease. It's the ratio change. You have more A beta. Usually you have a ratio from 40 to 42 from 9 to 1. And then it becomes a ratio from 1 to 1. So way more 1 to 42. So 1 to 42 is the way more immunogenic stimulus compared to 40. And so I think what you do is you're not enhancing the total A beta, but you're enhancing the immune stimulus for the microglial cell compartment.
Thank you very much for the presentation. It was really interesting. And I would like to ask if there is, I mean, the processes that are related to microglial activation are always in the same regions and in the same moment than astroglial reactivity.
I believe that microglia cells are faster in reacting because it's the astrocytic compartment or cells also express some, but not all of the pattern recognition receptors. In many of the tissues of our body, the immune cell component is faster in reacting and providing the first immune defense. So also that's what nature has made them for. The astrocytes come later. And we found in several papers where we used single exposure, single time points of A beta injections in rat and mouse brain that the first phase was a microglia phase, which was rapidly over. The second phase was an astroglial phase and it was sustained. It stayed for weeks, up to a month. So you can't say one or the other is more important. It's just a temporal and a spatial change. The microglia cells are the fast fighters. The astrocytes, they react in a more sustained manner. They also can produce inflammatory molecule, not to the same extent, but because there are so many more as compared to the microglia, it has the same effect.
Very interesting. Thank you.
Thank you very much, Michael. I think a very nice talk. Excellent. I am also interested in the locus coeruleus, but from the point of view of Parkinson's disease. Mm-hmm. Parkinson's disease, like Alzheimer's disease, also seem to start on the early degeneration happens in the locus coeruleus.
Yeah.
And in both of them, there is microglial cell reactive and also astroglial cell reactive as well. What are the common features that are happen in both diseases, and what is the key point or the molecular mechanisms that makes this divergent in a way?
Yeah, so it's a very, very good question, and it could be better answered by a pathologist. I don't know. I only know the answer from reading, not from doing the work directly. But if you look to the LC is like a very complex three-dimensional structure, and there are degenerations in various diseases. As you mentioned, the LC degeneration in the Parkinson brain is by more substantial and more profound as the degeneration of the substantia nigra. Most of the Parkinson researchers neglect that so far, and but it's a truth. So the point is that the area or the volume that degenerates in Parkinson's is the part of the LC that projects to the substantia nigra. The part that degenerates in Alzheimer's disease is the part that projects to the limbic system and to the neocortex. So the regions within the locus coeruleus are different. That's the primary difference. The molecular mechanism, I can only offer norepinephrine protection and norepinephrine control of microglia cells and astrocytes, but we know that this LC produces other neurotransmitters, and there might be further activities and mechanisms. But what we see that if we can use L3 DOPS, which is a clinical drug, they used orthostatic hypotension in Parkinson patients or pure autonomic failure patients, which are very few. But those patients benefit from this precursor of norepinephrine. It's been converted by dopa decarboxylase on the outer membrane of astrocytes and then turned into norepinephrine. And there's a very elegant Journal of Neurochemistry paper published in 1996 by Stephen Thomas, who showed that you can almost up-level the levels back to normal by peripheral treatment of mice with this drug. So if we do this in the animals, we regain control back of the microglia cells, and they do their homeostatic job again. So a big dimension of what we see is mediated by losing the norepinephrine control over the myeloid cell compartment.
Thank you.
An online question, and then we come to you, Theo. So it's from Isabel Lastres, and her question is, my question relates to the fact that the, how do you say that, NARP3 activation process also occurs in in other neurodegenerative diseases. Could ASC be the core factor driving protein aggregation in neurodegeneration?
Yeah. It's a very good question. Hola, Isabel. We tested whether ASC would be incorporated in neurons and template tau, and it didn't do in the first experiments we have been looking at. We never stressed the neurons. We just did it under physiological conditions and didn't see an uptake. Having said that, there could be that neurons also have their own ASC because there are four different, as you know, four different ASC-dependent inflammasomes, including NAB1. And NAB1, as shown by Andrea Leblanc in Montreal, is present in neuronal cells as well. So it could be that there is a role for ASC and tau aggregation. We haven't tested that, and I don't know data proving that yet. Sorry.
You have presented in your beautiful task the beta amyloid as the main trigger of the neuroinflammatory response and the microglial activation. And do you think that maybe all triggers, such as microbes or microbial products, as trigger of the inflammation?
Yeah, that's a very good question. First of all, I could not show all the data when you look to the paper by Christina Ising in 2019. We showed that the tau monomer and oligomer is a stronger inducer of the NAB inflammasome as the tau aggregate, and alpha-synuclein is in all the amyloid structures are basically inducers. It's always the same pathway. It's a highly conserved pathway. Why? Because it protects us from pathogens, or was designed to protect us from pathogens. And we discussed that before. In 1830, a German male would become 38 years old in average. In 1910, around the time Alzheimer described the case, a German male would become 48 years. And that excludes childbirth death. So not unrelated to that. And people would die mostly not from cancer, mostly not from cardiac infarction, not from stroke, and not from dementia. They would die from infections. So over a million of years, and through our recent evolution from the Neolithicum to our days, the immune system has been the key event in making us survive. And that's why it's so important and so profound. But now, since we doubled our lifetime in only 100 years, which probably no other species has achieved on this planet before, we now pay the costs of that. And now in the later ages, where we have accumulating of these beta sheet structured amyloids in our brain, we see the immune system reacting to that, because over a million of years it has been learned that's exactly what it has to do.
So there's another online
think-
question. There was another online question before. Then we come back to you. That's about the relation between ApoE4 and the activation of the NAP3 inflammasome.
Yeah, very, very good question. I wish I would know that. I have a very intense discussion with Dave Holtzman, because he says there's no deposition without ApoE, and I say there's no deposition without ASC, and maybe ASC and ApoE work to our brothers in crime. This is what we test by now. What we also test is we're currently generating a lot of cell lines on the ApoE 4, 3, and 3 background, and we want to investigate whether that makes a difference in S-spec release and inflammasome activation. But these are experiments which are ongoing in the lab.
Hi, beautiful presentation, very nice data. I would like you to maybe elaborate a bit, thinking on, you're putting this evolutionary perspective. So I'm thinking more on a cross-species perspective, because of course, microglia in human is not the same as microglia in mice. So what are, if there are, differences that are crucial from a translational perspective, or if they are sufficiently, the different are sufficiently small that are not relevant for translating the mechanism from the beautiful mechanistic experiment in the mice to the human, since it's a very big jump in order to reach the patient with drugs and this type of thing.
Absolutely.
I want you to elaborate on this, on that big jump.
Absolutely. I tried to say that, maybe I was not explicit enough, but so in general, there's a very, very good PNAS paper published by Suk and colleagues, a Korean group, in 2013. And that paper has analyzed the adaptive and the innate immune system between mouse and man under 3 different conditions: burns, trauma, and toxemia. And the collective summary of it is that 60% of the innate immune system are not conserved. So it's different pathways, which means you engage the same pathway, but you get a different result. Of the adaptive immune system, it's 70%, meaning that you have 40% and 30% which are conserved. For this system, for the NAP system, it's highly conserved. It has the same structure, same sequence in mouse and in man. In every cell you look. It's one of the, that's why we were so confident, and that's why the companies are so confident. Still, if you look to the downstream systems, if you look to the nitric oxide synthase type 2 enzyme, NOS2, which is responsible for peroxynitrite and nitric oxide generation in the body, which is detrimental because it's a sustained effect upon inflammatory induction, you will find that in mice, the INOS promoter or NOS2 promoter reacts more to interleukin 1 beta. In the human, it reacts more to IL-6. But you have to know that what happens in humans is IL-6, IL-1 beta comes first, and the downstream effect immediately is the generation of IL-6. So you have one step in between. The response element is affected the same way. So that's how you have to really go step by step and signaling transduction pathway by signaling transduction pathway. For the NAP3, we know it is really highly conserved. Not only between mouse and man, you can go to lower species and or to primates, for example. It's the same thing everywhere. For other pathways, like the interferon response, things look different, for example.
Just last question, because I think the time is over. But I was interested in the therapeutic aspects, and then so in what stage is the development of these inhibitors, and where would you place this treatment in the continuum of Alzheimer's disease if you had good results?
Yeah, very good question. So they're very advanced. There have been clinical trials with the first generation of drugs, which were designed not to cross the blood-brain barrier. They were generated for gout, treatment of gout, and treatment of IBD. And so the next wave of inhibitors started 10 years ago, were then looking at blood-brain barrier penetrable drugs, and they now arrive in clinical trials. The first trial is ongoing in Parkinson patients. The data have been released at the Movement Disorder Society meeting in Honolulu in September, I believe, this year, and they look very good. Roche will move that into a phase 3 trial, and the Alzheimer trials are supposed to start next year. Yeah, phase 2A. Well, I always say as earlier as possible. Yeah, but no one listens to me anyway, so I will see. I can't answer that, but there's a very nice paper by David Kleinerman's group and Henrik Sederberg's group from UCL/Gothenburg, and they published a manuscript in Nature Communications last October, in October 24, and they could track the spec in the plasma of the patients. And so now we have, so to say, a readout parameter where drug activity can be measured, and combining the new drugs with that peripheral. So we try to develop cleaved-gasdermin D as a marker over years, together with cell signaling in Danvers, but we were not successful because we didn't get a matching sandwich ELISA to work, but Kleinerman had the S specs now, and I think that's a very good readout parameter. So we have a biomarker and probably a drug responsive biomarker, which is great. And together with the new drugs, that will help us to identify the ideal time point, and will also help us to see whether the drugs have been working. What I've shown you is that it protects synaptic plasticity, but it also protects spread of disease and tau phosphorylation. So I think it can be useful for a wide range of therapeutic meaningful processes. And that would tell me start as early as possible. If I would have something to suggest, then it would be early MCI at the latest. And I would go for people who do not yet have total tau raised above the threshold level of 450 picograms per deciliter, which is the background, so to say, and then look for the delta in tau. That would be my primary objective. I would not go for a clinical parameter. The first in a phase 2A study, I would go for delta tau and nothing else. We have calculated that we need 800 patients, and we would need 2 years to become really convincing. And the question is if somebody conducts such a long trial.
Okay, thank you very much. It was a great
Thank you.
talk.